laboratories and how you can get involved if you like. And today we're talking to Professor Goolam, and I will tell you that this series of sessions that we are running with GESDA, the main aim is to bridge our understanding of how science shows these predicted futures to shape the present, and how we can ensure that decisions and discussions on the future of science and technology include the voices of the African youth. We know that oftentimes we as Africans come last, and then we complain about why we were not involved at the beginning. And with these sessions, we are saying it's important to get African voices in, but most importantly, African youth, young voices in. GESDA, of course, stands for the Geneva Science and Anticipation, the Science and Diplomacy Anticipator. It is a Geneva-based think and do tank. And its mission is to anticipate, accelerate, and translate the use of emerging science and technologies to benefit humanity and mitigate their potential risks. So we use in these sessions, or GESDA, we use GESDA's flagship tool, which is the GESDA Science Breakthrough Radar. And the GESDA Science Breakthrough Radar, you can get it on the GESDA website. You just click there and you'll see the radar, and you can go through it. And through that, GESDA aims to anticipate scientific breakthroughs and to ensure that we use these predicted advances to improve the well-being and promote inclusive development. We don't want this development to exclude more people. We want this development to help us to close the gap, the inequality gap. And last week, we had an introduction to the radar, and we discussed artificial intelligence. And today, we will dive into another fascinating topic. As you saw the little paragraph that I wrote on my updates, the fascinating topic of human augmentation and neurotechnologies. And we'll talk to an expert, Dr. Goolam. But why is it critical to include the voices of Africa's youth in shaping the science of the future? Well, it's important because the youth can start working now to mitigate both emerging and long-standing inequalities. Not only by bringing diverse voices and ideas to the discussion, can we ensure we get the best thinking from every part of our society and corner of the world, but we need fresh perspectives in these dialogues and debates. And so this is a matter of intergenerational justice. As you, the young people in South Africa and on the continent, always talk about the importance of intergenerational justice. Young people will inherit the future, and therefore must be involved in shaping how science should be used to affect the future. So the vision of the Youth Anticipation Initiative is that through these critical and thought-provoking conversations, we ensure that we create leaders who tackle social injustices by training and upskilling future leaders to be bilingual in both science and diplomacy, addressing global challenges. So please stay until the end of this session. Of course, we want to hear your sessions, and Mubin will respond to them, but we also want to announce a challenge to submit ideas, your ideas, about how you can use the future to shape our present based on any of the topics we have covered in the webinars. The webinar last week, if you missed it, it's on my timeline, on my Instagram timeline, get it there. So either the topic last week of artificial intelligence or the topic this week. The prize is an all expenses paid trip to attend the GESDA Summit 2023. This is a global summit of thought leaders on these topics that will take place in Geneva, Switzerland, from the 11th to the 13th of October, 2023. The summit will bring together academics, politicians, diplomats, executives, and investors from around the world, and of course, you, young people. So I want us to start with an introduction to the topic of human augmentation as covered in the GESDA Science Breakthrough Radar by GESDA Science senior advisor to the board. And our senior advisor to the board is Dr. Gerard Etcher. Gerard, are you there to give us some introduction on the topic of human augmentation as it's covered in the radar? Over to you, Gerard. Welcome everyone on the platform. Welcome. Over to you.
Thank you so much. And this looks like a loaded with energy meeting. We need that. Also, that is also neuro augmentation. So why are we concerned about human augmentation? Specifically, I think we will deal with neuro augmentation, with brain augmentation. Of course, we can augment muscle and digestion, but what we are really fascinated with is our capacity, our cognitive capacities. So we think that we live in a super interesting moment because all the neuroscience is now moving by and by out of the lab, and of course to strange places. It goes to the clinic where it should go because we still have huge problems from Alzheimer to paralytics, to paralysis, to epilepsy, and to mental illnesses in general, which are all or most of them really poorly solved and treated. So it moves to the clinic in a fast pace, but it also moves to the military. When you have a 500 million plane, you don't want the pilot to fall asleep, so you would like to probe his brain so that he doesn't, that you can read his state of alertness and so forth. And it moves to the consumers. And we think very soon it will really be a question to everybody, as parents, as young, as workers. Do we want these technologies to augment our capacities? So we specifically speak in a 10 to 25 years frame. Today, when you hear breakthroughs like, now we can listen to the thoughts of a person, this means that it has happened in a lab, and the training took months. It was with one person, his or her specific signals in the brain would not be transferable to another person, and it took hours and hours to work so that we can read the signals that the brain produces when the brain thinks or sings or hears music and so forth. I will go into specifics. So today, there is not a danger of that someone in secret is reading your brains. It needs a full cooperation of the patient, a patient who, by the way, has his brain stuck with electrodes, so he knows that something is going on. But in the future, this will be wireless. This will be different. This will be a helmet that you wear under your hair or on your hair. I don't know exactly. And it will be very small and inobtrusive. So we want to be prepared and to have the right ideas, the right regulations, the right questions that we can pose to these things in the coming 10, 25 years. We speak about augmentation and not correction or restoration, as we say normally. So restoration, of course, is done. It was just, so I have glasses, so my vision has been restored by a device. But today we go much further. We have now devices where we can restore speech, language, and so forth by people who were locked in, who did not move, who don't move their lips, who cannot write, who cannot speak, we cannot hear. But they have thoughts, and we can pick their thoughts with the right instrumentation. So we go further. So that is restoration. They spoke, they were paralyzed, they can speak again through a machine. The machine speaks for them, but it's their thoughts. So that's the miracle we can have today. But tomorrow, so that's restoration. which generally has already good rules in societies. The problem is equity, is the price, is the access, and of course the safety. I mean, does it work? It should work. But it's not something that is strange and will change our human nature. You know, we really try to help people. We have the walk again case a couple of months ago, you know, one person who was paralyzed, a combination of electrodes in his brain where they found a few good neurons still that had not been destroyed by his illness. And then machine learning, so AI that you heard last week, was the guy can walk again, this person can walk again with the help of reading in his brain when his brain is walking in, you know, when it makes the walking program, the electrodes in his brain pick it up and send through cables that we put in that body, send the message to the muscles of the leg, and the person can walk. And it's possible because of AI, because what didn't work until today is our brain is not doing one thing only at a time. It's a multiplex. It has lots of things. So it sees a beautiful, handsome boy or a beautiful girl, and it walks at the same time. So you have to do the things. And with machine learning, we learned to separate the things. So now the electrodes only listen to walking. They don't listen to the other distractions that the brain is continually usually doing, because it wants to see the whole environment all the time. Okay, so that's restoration, and I don't think it's just a problem. Really, it should be there for everybody. Yeah. Now augmentation, not yet there, but it will be there. And augmentation can take, it's a blurry line. If you talk to the deaf community and you say we can restore hearing, the community might say no, because with sign language we are actually, you know, we make do, we are happy, we have a culture, we have our things. So restoring also can be very close to augmentation. You know, really you push it to another level, you change something. The augmentation, of course, that is coming, and we will have to deal with it, and you will have to deal with it, is when we move to superhuman functions. If we augment old people so that they still, you know, react and have cognition like when they were younger, it is an augmentation, because aging is a normal process. It's normal that at my age I'm slow and stupid, but maybe they took a picture my kids will have at the age of 40, they will have a picture of their brain, of their cognition, and you can store it when they are 70. So it's a restoration, but it's also an augmentation, because the old guys will then act like the young ones. That will be a complication for society, because the old guys already do a lot of damage, I guess, or have done. But that's what. But it's still not superhuman. So it's an augmentation. But then the superhuman is, we know the genes for infrared vision. Humans do not have infrared vision. But with genetic engineering, we can give that to humans. Of course, we are not in, I mean, I'm not interested, but the military is, of course, you know, then at night you can also do your job. You know, you can continue days and night. Or the perfect sound, you know, we can give everyone, we know what's the perfect pitch. It's a gene. We know the perfect pitch. We know the genes for better short-term memory. That will be a question to parents. Do you guys want to go to the University of Cape Town, or just stay at home? But if you want to succeed in Cape Town, maybe an augmentation will help you, so you are better at exams if we give you a push for the gene for the better variant of your gene for short-term memory. So augmentation will be really a societal question. So it will not be just a problem of access. It will be a problem of choice. Can we build a society where we have the choice to do it or not to do it, to opt out, to go back in, and so forth? And what pressures do we think are acceptable? So it's the youth, that will have to deal with these things in about 25 years. But for your kids, there will be these questions. Do I help the guy? You know, Do I make him learn German day and night, or make a little augmentation so that he can get the German much easier? So today it's theoretical, but we have the tools, or some of the tools, to do it. And they are in the possibilities. GESDA is not saying that is a preferred way, but we think that the community, the young people of Africa and elsewhere, but the community as a whole society should be aware and should start to take steps to have the best implementation of these technologies. So maybe, Marga, you are the boss of time. Should I still, do you want more? I can give a few more examples in the radar of neuro augmentation, but I think I did the first shot. Or Getty, what do you think? Should I be a little bit longer?
I think we should go to Mubin. Let's
exactly.
Mubin.
So yeah, let me finish with the I think the technique. I mean, the things where it will happen are in the brain-machine interfaces. I alluded to those things. You know, when you make people walk again, it's machines. That's the brain-machine interfaces. The second technology, which will make a difference, a huge difference in the future, you will hear from Mubin. It's organoids, because these are brains made in, not brains. These are structures made in the lab that we can use for many ideas. And the third one is genetics, as I said. So genetics with the CRISPR and follow-up technologies. We will be able to be very precise on how we modify human beings or animals, for that matter. And the last part is the computers that mimic the brain, that become closer and closer to the human brain. Today they are far away. They do the things very differently, but they will be in 25 years much closer, and we will be able to build a computer that is much better than the human brain, because we can just construct a silicon brain with many more neurons, many more precise neurons that we will know how to do it, many more neurons that we have. We have about 86 billion, so it's not bad, but we can make a computer with 10 times as much. And then God knows what that guy or what that computer is going to do. Probably just wants to play golf, but we don't know.
Yep. Thank you very much, Gerard. I can see Ayanda already with the comment that says, wow, 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 this would require so much regulation. And she's asking, who would be responsible for deciding what is acceptable and not? I find that I'm more scared about this than excited. Maybe I'm wrong. Well, Ayanda, stay put, because we can talk about that. Of course, that's the reason why GESDA exists. That's the reason why this work is done. And we are delighted that we have Dr. Mubin Goolam, who's doing it in South Africa, so that we as the African continent can start engaging on these issues. Let me tell you a little bit about Dr. Mubin Goolam. Of course, he's a UCT graduate. He did his undergrad and MSc degree at UCT, and then he moved on to Cambridge University as a May Gray Fellow in Physiology, Development, and Neuroscience, and then he went on to Oxford at Wilson College, where he had a junior research fellowship. And now he's back at the University of Cape Town, and he's opening the first brain organoid laboratory in Africa. And we'll ask him about it as we go along. But Goolam, thank you so much for joining us today. Thank you for making the time. But most importantly, thank you for choosing to do the work that you do. And because you are allowing us through your work to inspire young people on our continent to participate in the future of science. You are working to establish the first brain organoids laboratory in the African continent. But before we get into that, let's start with you telling us about your journey. What got you interested in this field, and how did you get to where you are today?
Thank you so much, Professor, for such a lovely introduction. It's so nice to be. I wish I had an organization like GESDA when I was going through university. It's superb hearing about your work, and it's going to be so vital moving forward, as we'll get to that. incredible question Ayanda asked, really on the ball. So what got me interested in my field was when I was still in high school, I read this incredible article in Time magazine. It was on the front cover, and it was about stem cells. So stem cells are these cells that have the ability to change or differentiate into any cell type in the body. So we can take a stem cell in a lab, and if you poke it just enough, it turns into a neuron. If you poke it in the other direction, it turns into a piece of liver or a piece of heart. And I thought that was fascinating. The fact that they can change to any cell type, and they were going to be the future of medicine. So I really wanted to study about stem cells. So when I went, as you said, I started my university career, and I really wanted to study stem cells. And then as I was going through UCT, doing my degrees, I realized there wasn't much of that kind of work happening here. So I had a really amazing opportunity to study overseas in the UK, Oxford and Cambridge, and learn more about and hone my technical skills about stem cell research and making organoid, and I was amazed by it. And then at the end of this period of research in the UK, I had this choice. I could stay in the UK and I can try and build my research career there, or I could return back home and bring this technology back to South Africa. And I thought back to when I was a student and I couldn't do what I wanted to do because no one was doing it. So I decided I'll start my own independent research group in that field that wasn't really existing as today. So I got interested in the field from reading articles in school and then studying through it all the way to university. How I got to where I was today, I think I was a good dose of fortune. I happened to get really robust funding from different corporations and different organizations, and through a lot of hard work and perseverance and interest. If you're really interested in something, you want to pursue it, then you really got to go for it, and I think you'll eventually get it. So that's a little short introduction of where I got where I was.
Right. So Mubin, and you grew up in Cape Town.
Yes, I'm from the Cape Flats. I grew up and risen to.
There you are, Cape Flats. I mean, you don't even come from a posh place, Cape Flats, and here you are to educate us today. But let's start with the opportunities of this groundbreaking technology. What are brain organoids? You are opening the first laboratory to look at this. What is the value of this research for disease modeling, new therapies, and drug development, particularly with complex organs like the brain?
So that's the excellent thing. So stem cells, as I said, have the ability to change into any cell type, and organoids are ways that we use stem cells. So if you take those stem cells that can change in any way and you grow them in three-dimensional environment in the lab and you manipulate them, they'll form little clusters that will self-organize and turn into functional cell types that can recapitulate some of the structure and function of an in vivo organ. So scientists have taken stem cells and turned them into little heart-like organoids, which can beat like a normal heart. They turn them into liver-like organoids, which have the enzymes and the molecules present in the liver. And so it's a very powerful technology. We can make little structures in culture that represent an organ in the human body. And the reason that this is so critical and so valuable is that we can use these to study diseases and disorders. It's very difficult to study disease and disorders when they're in people, but if we can make a little model of an organ and look at how these organs interact with different drugs or interact when they have a disease, then we have a really robust way where you can really influence how the disease studies works. So brain organoids are exactly that. They're a tiny little model in culture made from stem cells that have functional and structural similarity to some parts of the human brain. And the value of them is absolutely enormous because the way you study the brain is quite challenging. So you can talk to patients and study diseases in people, as I say, but it's really limited and too late to help. And of course, there's ethical considerations. You can't do experiments in people to try and find out how to cure a disease. You need to do that before then. So what classically the way of studying diseases is to use animal models. We studied the brains in little mice or little rats. But unfortunately, rat brains and mice brains are very different to the human physiology. And of course, there's a huge ethical consideration of using animals too in research. We need to reduce our reliance on animals and find alternatives. And that's what an organoid is. It's made from human cells, so you can make a human-like model in the lab. You don't need to rely on animals, and you don't need to do anything potentially risky to humans. So you have the best of both worlds in many ways, and you can robustly study different types of diseases and things that happen in the brain in these little models in culture.
Wow. And Mubin, I mean, what about Africa? I mean, with all this potential and promise, I was shocked to learn that brain organoids have never been developed from cells of an African origin, despite the immense genetic diversity within African populations and the significant role this can play in diagnostics and treatments. Why is this important, and what challenges are you encountering as you establish the first African brain organoids lab?
This is the absolute key for me. I think this is the key moment in sort of my career. I had established myself at this world-class university overseas, and I was ready to go, and I was like, I want to build my independent lab, but I want to study African diseases. So I'm going to do it here, or should I do it at home? And that's what I decided. Like, if you want to study Africa and improve access to technologies in Africa, you need to do it in Africa, because no one in the Northern Hemisphere, quite rightly, was going to be interested in making African brain organoids. And not want to use African stem cells. They're in Europe. They would want to use their local technologies. So if you want to, if you're from the global south and you want to do the research, it's so important to do it here. And it is so significant because we are the most genetically diverse population in the world. Africa's this homogenetics. Everything started here. So we have such diversity that our way diseases and genes interact are going to be different. Studying them in that context is so valuable. So let's study the African brain with African cells in Africa, and we can tell how drugs affect the African brain and disease progress on the continent. So as for the challenges, I'm still in the very early stages to developing my lab. I only started my lab during COVID in 2020, so it's been a rough, fierce few years. So making brain organoids is not an easy process. It sounds I make it sound simple. You take a stem cell, you make an organoid. There's a lot of steps in between. But the challenges being faced here are the ones that I think we all face in many ways. So the major one is lack of resources, particularly funding. We don't have robust funding in Africa to develop large scientific protocols like they have in other places in the world. And a lot of the reagents and the items that we need to make brain organoids were developed in Europe or in Asia, so they need to be transferred here, and there's lots of costs involved. So there's few opportunities funding-wise sometimes to build them. The other important thing is that there isn't a capacity, which is the one thing that bothered me, that we don't have a capacity, the space, and the sort of expertise to do this type of research. But the way we build that is by what we're doing now, is that it's novel in this landscape, so we have to build capacity from the start. So building from a start, as opposed to walking into a situation where things are already established, is a big challenge. And that's what we face in Africa. When we build capacity, we have to start building the capacity. But what I will add is, when I was thinking about sort of the importance of capacity building, the one thing we don't lack, that we really have here, is intellectual capacity. The students I encounter every day in all my classes, and I'm sure you'll say the same thing at UCT, are just as brilliant and excellent as all the ones I've seen at Oxford and Cambridge. The scientists here are brilliant. So that gives me hope in the building. It's not that we're lacking the ability, it's that we just need a bit more time to grow and develop, and a bit more investment in certain areas, and we can be right there.
Yeah. People are asking amazing questions on the platform on Zoom, but also on Instagram. Just to let you know that I will take your questions. I'm noting them, very interesting. Just stay on. I am at some stage going to pivot to your questions so that Mubin can engage with them. But let's tackle the risks, Mubin. There's a concern that neurotechnologies could lead to enhancement in healthy individuals, and therefore raising ethical questions about access, equity, and the impact of human character, safety, and freedom. There's also fears about brain hacking, where the ability to read and write into the brain could be misused to manipulate human minds and behavior. For example, in the military, And this could pose ethical dilemmas related to augmenting soldiers' combat abilities. So some have called for a modification of the Human Rights Declaration to include neuro rights. How do you think about regulation and governance in your work? Should scientists themselves define, or there should be no regulation at all?
So this is a great question. I guess this is why organizations like yours exist. So the first thing I just want to qualify, because I think talking about brain organoids to the audience might sound quite scary. So let's first differentiate, and Dr. Esther said the same thing, that an organoid is not an organ. It's not the actual thing. So brain organoids are not a human brain. They can't think, they can't feel. What we use them are as representation.
You keep disappearing. So they are not a human brain, they're an augmentation.
No, they are representation of the brain, not no. Yes, sorry.
They're an augmentation of the brain. Okay.
A representation of the brain.
Okay.
Yeah. So they don't think, they don't feel. When we use them to study certain aspects of the brain, they are a model. They are no way actual brain. So that's an important thing to note about this specific technology. So, but then let's talk more about your neuro rights, because that's really important, and it brings into where we fit and what exactly this topic is about today. So we need to define that all of these studies and the research we're doing is specifically preclinical, before you get into humans, disease and drug development. And the way you do that is you define the limits. And should scientists define by themselves? Absolutely no ways. Scientists shouldn't self-regulate what they do. This needs to be, we don't exist in a bubble. Its potential consequences of any technology need to be considered. And it needs to be an open discussion with all of the stakeholders that are involved. So yes, that includes us scientists, but government and religious groups, group advocates, community leaders, anyone who has a potential say in this needs to come together and discuss through organizations like yours. That's why they are so important and should be around. To discuss together to develop the correct documentation to govern how work processes. So I think a scientist's role in this regard, you provide appraisal of our technology, what it's used for, its potential to be used for in a good manner, and then we discuss how to control and use good and not in a damaging fashion. It's creating an appropriate boundary to allow technology to flourish and help us without allowing it to be predatory. I think it's a very difficult balance, but it's the conversations we start early. As this is the whole point of GESDA, you start the conversation now before the technology has the ability to do anything damaging, so we can set up the boundaries so that we can still do our research and find incredible things to help humanity without leading it down those scary topics that we just discussed.
So it's a good thing that we are discussing this now and we're getting young people on board, because it's important to think about these ahead of time, and they can ask the questions that we might not ask when we're sitting in a room together as adults. But how do we ensure that these new technologies, such as the one that you're talking about, do not perpetuate inequalities in Africa? We don't want them to worsen existing social disparities or create new ones. How do you hope, Mubin, to make these technologies accessible more broadly while balancing potential risks?
Yeah, you know, this is a question I've been asking myself for quite some time. Is how do we make sure the inequalities in this continent don't keep existing? I think the way is to develop things in Africa. If we want to have access to things, we need to develop our own capacity. It needs to be driven by locals in the continent, by Africans for Africans. I think is the only way we can do that. And I think what we do it is with equitable relationships, and that's really important that they're equitable with funders and global North stakeholders, where we are equal partners and not subject to a predatory type of collaboration, because often those collaborations can be predatory and they really kill long-term capacity building. So as an example of an area where Africa is the world leader, I want to think about the research in terms of infectious diseases in South Africa, and really in South Africa in particular, is that over a long period of time, there's been a long-term investment to build local capacity in tuberculosis, HIV, and malaria research, because there was such a significant need in Africa. Local heroes, local experts were the ones that were discussed with, and they built the places to do the research here. and a massive global investment and equal partnerships. And now the best place in the world to study those diseases is right here in Africa, because we have, that's the model system. We build capacity in that way. And to mitigate risks, I think it's dialogue that does this. It's discussions. It's open and honest, frank discussions with all the stakeholders. Things that we're doing right now is we develop regulations that support research without inhibiting the progress and prevent exploitations, which is a fine line to balance and easier said than done. But I think it starts with opening the floor, open the conversation, get all of the youth involved in the conversations, because it's them who's going to be affected. Set limits and regulations early in conversation, and be open to changing your mind when things are important.
Great. And Mubin, how can young people get involved in this exciting work? I mean, the GESDA Youth and Anticipation Initiative aims to engage and empower young minds to contribute to this revolutionary field. But how can these young people who are joining us right now in our audience, who are online, there's like, 100 of them, how can they get involved in your work? Is there a way? How are you working to train the next generation of researchers in Africa in this field? Is there a team that you're working with? Are there sessions that you hold that you might want to tell our young audience about?
Yeah, I think, if you're interested in someone's research and you hear about them, is get in touch. If you're interested in the work, you're very happy to get in touch with me. If someone's interested and you want to hear more about them, I'll tell you that most researchers and scientists across the country, in particular in South Africa, are more than willing to discuss research and get involved. The way to do it is get involved with our organization. So I'm part of the Department of Human Biology, and there's incredible neuroscience institute at the University of Cape Town, and the Neuroscience Institute and the department are more than willing to have you come and visit us and learn more about our science. So in particularly, come to UCT if you're interested in my work, study our degrees and get in touch with us. Especially if you have a biomedical degree and you want to study further, there's so many options available and funding avenues available to you. So if this kind of research,
Go ahead, Mubin.
If this kind of research interests you, or if you know about other research, if the research from the last week's AI researcher, get in touch with it. I think most scientists love to hear from young people who are curious about their work. We are curious about our work. We want to tell people about it. So that's how I'm building. I have a lab. I take on the best that this university has to offer, which is the best in the continent. And we teach them about stem cells and organoids, and they become stem cell experts and organoid experts themselves. And that's how we advance the field.
Mm-hmm. And Mubin, do you have students, for example, who are doing psychology, interested in brain research, who may join? Would they join your group, or would they need to go to another group in the humanities?
So I'm going to again talk about the neuroscience. At the Neuroscience Institute at UCT, we combine clinicians who are neurosurgeons, as well as psychologists and psychiatrists and basic cell scientists. So psychology is not really my expertise, but if you're interested in psychology and learn more about the brain, you can get in touch with us at the Neuroscience Institute, and there are people there who will be very interested to hear from you. I think that's the best way to go about it.
Great, but I'm trying really to get to the issue that it is really an interdisciplinary kind of study. I'm sure I can see people from mathematical sciences who might be interested in working in a team there. They can be useful in some research group doing brain research.
Absolutely. We've got to have crosstalk between our various disciplines. We too often sit in silos in academia within ourselves. Absolutely, we need a more crosstalk across the various fields. I think that's what common spaces like the Neuroscience Institute really govern. They give you space to crosstalk between each other. But yeah, please get in touch. If you're interested, you always can come and chat to me.
Fantastic. Mubin, I'm going to spend about 10 minutes taking questions from the audience, and I can see Tsulu asked the question soon after we started. Tsulu, are you in the audience? Do you want to tell us your question, or do you want me to read it? Tsulu? Tsulu is not speaking to me.
Please read it, Prof, if you can kindly read it.
Great. So Tsulu's question says, how can neuro augmentation assist the neurodiverse, and in bracket, autistic individuals?
Wow, that's a great great question. You know, I think that I don't have a really good answer for you, Chula, and that's because I don't know enough about the neuroaugmentations that would be appropriate for an autistic individual. What I could tell you out of interest is that one of the aspects we do with our brain organoids, which I will distinguish from a neuroaugmentation, so I'm not answering your question directly, but I think it's one of the tools we use to study autistic brains and the genes that are affected in autistic brains. But how a neuroaugmentation could assist a neurodiverse population, I'm gonna plead that I'm not an expert in that topic individually. A really great question, but I don't actually know. I have a proper answer for you.
What
Yeah.
about stem cells, if I may interject, Prof?
Yes,
what about
please.
stem cells? Because I heard that you also looking at the stem cells. Is there do you have any experience with stem cells being used for autistic individuals to sort of augment?
Yeah, that's a really important question, and I want to, because there's a lot of talk of that kind of thing, and that's one of these things where there's a limit and there's a boundary. So stem cells used as a treatment should not be something that we are doing. We are using stem cells to build models. We've not talking to patients, not influence them on patients, but to build them on to understand how disease progresses, not to transplant or use them with patients. That's a really important point to bring up. Stem cells transplanted to patients, it's a dangerous kind of field that in some parts of the world is done, but is not something that needs to be very tightly regulated, because it can be very, very dangerous. So it's not something I believe in. I don't think stem cells, I think it's too dangerous to try and use them as a treatment option, but rather as a model of study, if that makes sense. That's a really important point. And that's where regulation is important, because there's really damaging things which I talk about in some of my lectures to students in medicine, that in parts of the world where they use stem cells for treatment, that it can lead to very, very dangerous effects.
Yeah.
Great question.
True that, Mobile. You know, my son has been dealing with cancer, multiple myeloma, and he did stem cell transplant. I mean, very dangerous, you know, could have died, you know, thank God he didn't, but the transplant, he was a self-donor. But they generate stem cells and do that, exactly what you're saying. It's quite a key kind of a procedure. Thanks, Mubin. Ponzo, I see your question. And Pumza, I saw your hand. I will come to you after Ponzo. Ponzo Mushokwa, do you want to ask your question? Okay, Ponzo Mushokwa's question says, could we talk disability? Can this research make an impact in disability?
Yeah, absolutely. I'm loving these questions. It's a real great getting to good topics. Absolutely. So that's a wide range of things. But I think we'll talk about, if you talk about like neurodisabilities, again, I draw a distinction between treatment and studying disabilities. But if you model the brain and how it works, then you can model when damage occurs. So if you lose function in certain parts, you can model that using organoids, then try and understand which pathways and networks are broken, so we can think about ways to fix them. So great correction point. So there's a huge field of studying how different kinds of disabilities and traumatic injuries to the brain can be affected in organoids. Excellent question.
Right. His Excellency Prince Gift is appreciating the lecture, and he says he has never learned this much in such a short space of time.
Oh, that's
Mubin.
That's
Pumza,
beautiful.
give us your question. Pumza.
Thank you so much, Prof. So it's not so much a question, more of a comment. I see that this topic in particular is multidisciplinary. I think because it also, I'm in education, so it fits very well in there. I think the best way, from my perspective, of how this project in various ways can be implemented throughout Africa is through using participatory action research. So with it being multidisciplinary, people from education or biology, neurosurgery would come together and identify a problem. How do we go about solving it, working together in different in different disciplines? I think if it's something that can be birthed in Africa, it could have such a huge impact just throughout the world, because we'd be working with people who are directly impacted and affected by this, and they would be participating. So they would be co-researchers instead of subjects or people that you would be using a certain chemical, whatever the case may be, on, but they would be willing participants. So I see just the potential beauty of this, and the mere fact that there are so many disciplines that are put together that can work collaboratively here. I think participatory action research would be the best way to implement and just reach throughout Africa and the world. Thank you so much. Thanks, Prof.
Thanks, Phumza. Phumza, I'm going to advise you to wait until the end and hear Marga describe the challenge. Who knows? You might come with a creative answer to the challenge. I see Prince. Well, before we go there, someone asked Mubin on Instagram, where do the stem cells come from? The human stem cells. Yeah.
Excellent question. This is absolutely superb. So that's a perfect question. There's various places where you can get stem cells from, but about 10 years ago, so it's a very new technology, someone discovered if you take a piece of human skin and we apply the right different factors to it, we can turn it into a stem cell. So we can make stem cells from human tissue. So that's what we do. We get skin fibroblasts from the skin and we turn them into stem cells. So that's the simple answer. It's a long process, but we can create stem cells from human tissue, which is why it's so incredible and why we can start doing this from populations in Africa. Great question.
Great. And Hunadi Malika, Hunadi, you are on the platform. Do you want to ask your question? Hunadi is not there, so I read the question. Definitely exciting, thinking about all the incredible impact it can have to treat children with psychiatric conditions. Imagine how quality of lives can be dramatically improved. I'm just wondering, firstly, the ethics, and secondly, access. How will this be accessible to everyone despite economic resources? Is it even possible?
So firstly, for the ethics, it works as per any other project where you're doing work with human tissues. Is we have robust constitutional law in South Africa, particularly regarding the usage of human tissue. So we need to put together a proposal and discuss exactly what we're going to do, and we're governed by the government law, and we go through an ethics application procedure to make sure what we're doing is reasonable, not overextending the boundaries. So that's how ethics applications work in countries like ours. So that's a really important question, and that's how we go about it. And then secondly, access, that is a terrific question. Of course, that's the whole problem. That was one of the problems that I had when I was studying, is that I didn't have access to the technology at home. I had to go overseas. And the way we access is what we're doing now. We build capacity, Hunadi. We start from scratch. We build capacity. We partner with Global North. As Phumza was saying, you have to have proper partnerships that aren't predatory, and you build and you develop. So we develop our own capacity so people can have access to things that will otherwise only be present in the Global North.
Great. Jeffrey Lee. Jeffrey, I can see your question. Jeffrey, do you want to read your question?
For a really good presentation. So I just had a broader question, not on organoids specifically, but more on human augmentation. So we think that like human augmentation, if it's broadly accepted, how would it change military doctrine, right? So military doctrine here would be defensive, offensive, and deterrent capabilities. But when you have an overemphasis on offensive capabilities in terms of human augmentation, you could then lead into this Cold War kind of scenario, right, between countries that compete for this. And I don't think this would be limited to the quote unquote like great powers of the North, but any country that has robust bioresearch capabilities, right, which numbers in hundreds of the world right now, basically. And you have private and academic industries as well. So in your experience, like what are the ethical and legal bounds of applications of your work and human augmentation in general to the use in war, but also considering germline and somatic cell editing, where right, human germline would be carrying on for multiple generations and somatic cell line would end with the user at the end, right? Thank you.
Yeah. Great question, Jeffrey. So I think this is exactly why we need to have really robust and really proper documentation in place. So absolutely, in no place in the world, in north or south, can you make organoids and use them for augmentation. It's strictly not allowed. I'm sure there are parts of the world that have less rigorous doctrines that allow that, but no, you should not be able to, and the legal boundaries should be very strict in that regard. Particularly, you mentioned an interesting point about the human germ identity. That is a dangerous kind of territory, and I can only speak about my own personal opinion, which is that's not something that I think we should be entering into. I don't like the prospects of it. I think the potential negatives outweigh the potential cons, and that's a whole other different sort of kettle of fish, which is very different, of course, as you must mention, somatic cell editing. So I think there are rigorous laws in place, and we need to ensure that those laws and the lawmakers and the changes that occur, we all involved in those conversations. They don't allow for crazy augmentation for war type capabilities, because that is unacceptable usage of potentially very useful technology.
Great, thanks, Mubin. I see a question from Mitera. Mitera, who's at Nestle Care. Mitera, do you want to read your question?
Yes, Prof. Thank you so much. Firstly, thank you so much for hosting this workshop, and thank you for educating us. So my question lies around, you said that essentially what you do, you create models that are representations that help you study diseases and so forth, right? When you are creating this model, does it differentiate between an adult model versus a child model? Yeah.
Oh,
I
that's
my question is understood.
Yes, no, it definitely is understood. Prof, I'm so impressed by all of these questions. They're asking brilliant questions. That is an excellent question. So when we make organoids, because we make them from stem cells, because stem cells represent the earliest stages of life, the model you make starts off being as a child model. So what we're making is essentially a childlike brain. So you do all the model systems normally you'll hear about and you'll discuss, and if any of you hear about them, they represent an adolescent type of that organ. And then what we need to do to make an adult-like model is grow them and keep them going in culture so that they actually grow and develop and age naturally. So all stem cell models start off with being childlike, and we can grow and develop them into adult-like models, which is a real subtle part of the technology that you've tapped into immediately. Excellent.
Fantastic. Mubin,
okay. And
go ahead.
No, I think my last question would be an addition onto that. It's when you saying that you grow them organically. If you would like to study a 29-year-old, it means that you'd have to grow them up to 29 years. Would you have to wait that long?
Excellent. Another excellent question, and I didn't specify. So they don't, because they aren't the actual organ, they don't grow and develop at the same rate. So they don't reach puberty after 14 years. What happens is they in the first few weeks, they only live for a few months. In the first few weeks, they're adolescent, and they rapidly develop an age. So after a few months, they are more representative of an older brain. So the timeline aren't the same. It's you can go them for up to a year and a half, a brain organoid. And by the end of that period, they represent a very older brain. So the timelines are not the same. Great follow-up.
Great. Mubin, I want us to ask one last question, but just to acknowledge 2 people on Instagram. There's one person who's appreciating it. Their name is Lindela. They say, thank you for your honesty in responding to the questions. Thank you so much, Mubin. And then there's Prince Ntoni. Prince is one of the young people who won the opportunity last year to go and attend the GESDA Summit in Geneva, and he's made himself available. So Prince, slide into my DMs. Prince wants to tell you about their experience, and I can chat with her perhaps sometime about them, then they can tell you. I want Maga to tell us about that. And then Mubin, just the last thing, it's a quick one. This one is from a student who is at the University of KwaZulu-Natal, Sekema Fuka, who says, I'm a PhD student in neuroanatomy at the University of KZN. I'm currently working on traumatic brain injury. How can my work fit in? And that will be our last question before I hand over to Marga.
That's super. I'm very excited that you're doing a PhD in such a really cool topic. So that's a really good question. So as you'll know better than anyone, is that traumatic brain injury is a huge problem in Africa. So this is one of the things I want to use brain organoids to study, because how can you model traumatic brain injury and how that affects brain development and future problems is by, if we have a little brain organoid and we induce a traumatic brain injury, we could then potentially study the effects it has in a cell culture model. It doesn't require having to study them in humans. So that's a really great area that I would like to pursue in my future research. Very cool stuff.
Fantastic. Mubin, there's lots of appreciation and lots of questions, but we can't get through all of them today now. So I'm going to hand over to Marga, because I would like Marga to explain the challenge so that the young people on the platform know how to participate, because we need winners. Marga, over to you.
Thank you so much, Katie. Wow, this was fantastic. Thank you all so much for engaging with this topic and your excellent questions. Thank you, Mubin, for being so clear. This topic can be very, very complex, but I think you did an amazing job at really explaining
so much.
And the challenges and the fascination that you feel for it really transmits, and the importance to bring this research to Africa and engaging young people. So this is only a teaser. As Getty says, we have really a very short time today, but the idea is to get you all to think about this and get all these ideas and questions that you put in the chat into the Youth and Anticipation competition. This is an opportunity to come to the GESDA Summit in Geneva in October. And the GESDA Summit is a place where we'll precisely do what Mubin, what Gerard, what Getty is encouraging us to do, which is to bring all the stakeholders together, scientists, decision makers, diplomats, private sector, NGOs, civil society, all together to discuss these questions, because they do not have one answer, because they are ethical, they are political, they are research, they are business implications in all of them. And the GESDA Summit is where we discuss with all the stakeholders to hopefully arrive to an agreement on what should be done before the applications of the technology are out of our hands. So it is very important that you participate. We will be announcing the guidelines. I will tell you a little bit now, but basically we want you to take any of the aspects of the topics we've discussed in this webinar on human augmentation or the previous webinar on artificial intelligence, and those webinars will be posted on Zoom, so you can rewatch them and process all the information. And then we will have a questionnaire for you to fill out, a very simple questionnaire about how do you see these topics for your particular context, for your work, for your study, for your field. It's a very interdisciplinary field. So if you work in psychology, if you work in neuroscience, if you work on politics, you can have a different perspective on this topic and bring it together. So you will submit an idea. So this competition is very short, only 2 pages, on how and why this should be important for Africa, and how do we go about it, right? And then we will hopefully select a few of you to join Getty in I don't know if you're going to have them on your plane, Getty. I think you said that last week. That would be very cool, your delegation.
I'd like to go with them. I'd like to be in the same plane, and then we can
Exactly. And we'll see you all in Geneva, and then we'll have the chance to discuss with global leaders from all around the world, from all sectors, to make sure your voice is heard. So stay tuned. How do you hear about it? I'll just post it in the chat, a number of links. So the GESDA website, so GESDA News, GESDA Twitter, GESDA LinkedIn, and of course Getty will be posting on her socials and Instagram, Twitter, and all of that. So the next few days, please stay tuned, because we're going to be posting the guidelines and the template that you need to fill out and return to us. And I think that's all. Anything else that I need to say, Getty? I think we're good. Thank you so much, Mubin. And I would like to, before we close, to give maybe a word to Gerard, because a lot of the questions that have been asked, actually GESDA is trying to bring stakeholders together to tackle them. The question of, are scientists, can we self-regulate as scientists or not? So those questions that you asked, the question of what should we do about governance, diplomacy. So this is why GESDA exists. As Mubin, you have a guest. Gerard is here and wants to say a word, would be the way to close. Otherwise, Gerard is already gone. We'll leave it for the.
No, I'm there, but
okay, great. So Gerard.
No, so I learned a lot. You know, it's good to listen, and we will use this. We will use this in our reflection. I would, you know, as Mubin, you are this star scientist. So self-regulation is not that you make your own rules. Is we ask you where you would stop your work. What will you not do with brain organoids? And then that's your norms, and we held you accountable for that. So that's self-regulation. You said you would not do it, and then you do it, then you are punishable. You know, but you put your own rule. So it's not that it replaces rules by society, but you have as you have shown in your answers to all these questions, scientists are the first to know what the technology can do or not. And it's important to listen to them and to ask them, how far do you want to go? Where do you stop? What would you not do? And then we follow you. That's for me self-regulation. So it's science integrity. And it's very important for fields that start. You are the first one to do it in Africa. You know, you started. So you have this responsibility to show how far you want to go. That's the point. And GESDA thinks for exciting new feeds that we don't want to kill, because you are right. Your cells are juvenile cells. You know, they are baby neurons. So they are important for autism, because autism develops in those stages. You know, it's not an old people's problem. It's a young people's problem. So actually, it's great. So you have to do it. So, but you are responsible to say, that's where we'll stop. That's what I'm not going to do. And so long live la science. In Africa.
Thanks, Gerard. Mubin, you want to close remarks?
Yes, I just want to thank you very much for inviting me and having me here. It's really exciting to hear, but just I wish I could apply to that competition. I really encourage you all here to do so. It sounds like it's going to be a fascinating experience. So thank you very much, Prof, for having me. Thank you, Dr. Ashley. Thank you, Marga. It's been a lovely webinar, and thank you to all of you for attending and all of your really insightful, brilliant questions. I never get ceased to be impressed by students. I'm sure Prof will agree with me there. Every time you have a conversation, you just learn something new from people. So it's been fantastic. Thank you.
Fantastic. Mubin, thank you so much for your time, for sharing your work with us and letting us in. You've ignited so much interest. I can see young people on Instagram and on Zoom who are saying, oh, I wish I had, you make me want to change my career. And because they're now excited about what you're doing. So here's the thing. People are asking where to enter the competition. Don't worry, we'll put the link. Check my social media pages tomorrow. We'll have the link and the information. And in fact, we will go on live with Prince, who was one of the students, young people who won the competition last year, and then he can tell you more about it. Don't worry about the discipline. This is not just for people in biological sciences or in psychology or whatever. It's for, like Marga said to you, you think about your discipline and you think about the two areas. Watch the video on the GESDA platform or on Instagram, because we're recording, and think about, how do you use this technology? How do you think this technology can be used to resolve or to solve problems, challenges in your context or in your discipline? Or how can you bring your discipline together with this AI or human augmentation to solve a particular problem that you are experiencing in your community? Because you do not have to be registered with any university. You just have to be 30 years or younger. Then you can enter the competition. So watch out. I will remind you, it's important to put your best foot forward and as you enter this competition. So thank you so much for joining us. Love to see you here. Always fantastic. Get your other young people, your friends joining us all the time. So goodbye and have a good evening. Mubin, wait a little bit. We want to say thank you and give you a hug, proper. Thank you, guys. Grace.
Thank you, Prof.
Really nice to see you again.
Thank
His
you, Prof.
Excellency Prince. Thank you. Thank you. Thank you, Lebu, Remonelwe, and people on Instagram. Thank you for joining us. Lido Kushe, thank you for your support all the time. I saw also Tebogo Mabusela, the famous Tebogo Mabusela, also joined us. Thank you for joining us. Timothy Mboya, Karabo Tebato, Lebuhang Malefu, Mitera, Leonard. Thank you so much for joining us. And I hope you enjoyed it. Science is enjoyable, guys. Like after listening to this, go and check out other